Hi, everybody. This is Larry Johnson. I am a professor at Texas A&M University, and today we're going to talk about lysosomes, smooth endoplasmic reticulum, mitochondria, and inclusions. We want to look at lysosomal ultrastructure, their function and dysfunction, along with continued discussion of protein synthesis, as we started before. We want to look at smooth endoplasmic reticulum, this ultrastructure, some characteristics of its functions, and why it's so high in concentration in steroid-secreting cells. We also want to look at mitochondria, the ultrastructure, their function, the origin, and the incorporation of proteins inside both of those membranes, as well as inclusions, and we will end with inclusions.
Ribosomes translate messenger RNA to produce proteins. And here you can see ribosomes, some of which are in the cytosol, free in the cytosol. And those proteins go into peroxisomes, mitochondria, and in the nucleus. And today we'll see it in mitochondria. Also, ribosomes are associated with endoplasmic reticulum. And that goes to the Golgi to lysosome we're going to talk about today. Also, it's the rough endoplasmic reticulum that's associated with producing the membrane in smooth ER and enzymes in smooth ER and in secretory vesicles. So that's what we'll see today, as well as mitochondria. Now, there's two types of reactions in the cell. We have a scalar reaction, and that's what happens in the cytosol, where you have precursor A, precursor B, in the same package as a product C. In the same package. However, if you got membranes to partition cells, you can have a vectorial reaction. That is, you have precursor A and precursor B in a separate container than product C. And membranes allow you to facilitate that. And that's what you have when you have ribosomes associated with the endoplasmic reticulum, where the products are sequestered inside the rough endoplasmic reticulum away from the precursors, the amino acids, free amino acids in the surface. So we have both scalar and vectorial reactions. This is the free ribosomes, and this is what happened in the associated ribosomes associated with endoplasmic reticulum. Cytosolic or the RER proteins. Now, the cytosol itself is all the stuff between the nuclear envelope and the cell membrane that is not in organelles themselves. So it's all this stuff in between there. That's a cytosol. And among there, you would see the ribosomes out there making protein would be in the cytosol. And so here, free ribosomes are translating the messenger RNA, producing proteins for mitochondria. We'll talk about that today as a scalar reaction, or through the rough endoplasmic reticulum inside there for the plasma membrane secretory granules for a vectorial reaction, and lysosome, as we'll talk about it. Cytosol, RER proteins. So lysosome is the first thing that we want to look at. The ultrastructure looks very similar to that of any other secretory granule. However, if it's a secondary lysosome, which is what these are, you see varying intensities, very dark, light, lighter, dark, dark, and it's all in the same granule because things are being degraded. And so there's various states of degradation, and that's why you have the different colors there that we see in secondary lysosome. But a primary lysosome would just look kind of gray, like a secretory granule. And a lysosome has enzymes. It has phosphatases, proteases, lipases, lipid-degrading enzymes. It has a host of these inside there. And it has a lower pH because it uses ATP to pump in hydrogen ions. So these enzymes work better at a low pH, and they are in the lysosome at the expense of energy. Now, and the method of detection lysosome is localization of the enzymes in the lysosome. Otherwise, you can't tell it from a secretory granule, as I mentioned. So you use histochemical reaction that respond to the specific enzyme that's in there. So you feed the lysosome substrate, and it makes a black reaction product, which is what we see here, a precipitant, whenever the enzyme inside there is reacting with the substrate you provide. And here we can see that the Golgi apparatus is polarized, and so the mature phase is where you have acid phosphatase, and here you can see where acid phosphatase is pinching off the mature phase of the Golgi apparatus and is fusing with the acrosome of the developing spermatid. So a negative charge on the inside of it allows the membrane not to be broken down. So it has all the components to break down membranes, and so why doesn't it break out of its membrane? But it's the negative charges on the inside. I'm not sure exactly how they get those. That helps protect it. So we have primary lysosomes, and you have secondary lysosomes, ones that already started eating something. It could be something from outside, heterophagy, or it could be something organelles from inside, autophagy. And in either case, it'll be a secondary lysosome because it has the enzymes plus some of the things phagocytized to make a secondary lysosome, which has varying intensities of reaction product there. And here we can see the Golgi apparatus sorting proteins, and if it's got mannose-6-phosphate in there, it goes to the lysosome. If it doesn't, it goes constitutive for cell membrane proteins as well as secretions to occur, or it may go to a secretory granule that then may be receive a signal for these. This is induced secretion. This is a constitutive secretion. So this may be induced like a pancreatic astrocyte cells induced by insulin, inhibited by glucagon. And so hormone may induce these in this route. Lysosome mannose-6-phosphate is present. And so that shows you the cisternal, that is what's going on in here, versus the post-cisternal phase of secretion, and that's the sorting that goes on here. And here we can see what happens in the lysosome. The enzymes are there. You get a clathrin coat pinching off vesicles to transport them into the early endosome, and then the pH is lowered. Hydrogen ions are put in here, and the receptor separates from the cargo, and so here's your protein with a mannose-6-phosphate in there. The phosphate comes off, keeps the mannose with the protein, and the receptor is recycled. So you get recycling of the receptor. It binds at a high pH, and at a lower pH it dissociates, and the receptor is recycled. Now, a lysosome is associated with programmed cell death, like it's just a human skull, and you can see how it has to change with the facial features. It changes as it removes cells to build up other ones. And you know, of course, a frog starts out as a tadpole, and it needs to get that tail to regress and legs to develop as it transfers from a tadpole to a frog. The lysosome does that. A lysosome also has housekeeping function. Here we see a mitochondria and a peroxisome. Too many membranes. If you got too many membranes, that's an autophagic vacuole. Here's again, too many membranes around this rough endoplasmic reticulum, and so this is going to be autophagy, things to be digested. Here you see unprogrammed cell death, sometime associated with cardiac cells, is if you have ischemia. What happens is the cells think it's time to die, and they release their lysosomes, and that kills the cells. And here you can see my blood vessel. This is the circumferential coronary artery that goes around my heart, and this is before the stent. You can see it's about 90% occluded. but then they put the stent in there, and a great flow was able to come through after the stent. So thank goodness for modern medicine. So in summary, membranes allow a high degree of chemical heterogeneity. Internal partition of cytoplasm, substrate, products, the scalar reaction it allows versus the vectorial reaction. The vectorial reaction, it partitions things as we've talked about. Also, the membranes hold in reserve a large repertoire of unexpressed biochemical reactions. We're all sitting here with lysosomal enzymes inside the membranes. If it was released, then it would kill our cells. So unexpressed biochemical reactions are contained in our lysosomes. Whenever they chill meat for tenderizing meat, it's the lysosome that's self-digesting it. They even shock meat sometime to break away the membranes of the lysosome. So the function of the lysosome is autophagy and heterophagy. Heterophagy, where it's eating something from the outside, like say a bacterium. And the bacterium would go there, interact with the lysosome, become a secondary lysosome, or autophagy. So that's a function, heterophagy, autophagy, for digestion. Now there is a latency associated with lysosome because they're in membranes. So whenever the dude discovers lysosome, they discovered it. There was a latency in the characteristic. You had to pop the membranes to get the enzymes out, or just let it over a period of time. The membranes would pop, kind of like whenever you tenderize a meat by keeping it in a cooler before you cut it up. The end product of lysosomal digestion is a lipofuscin. So there are some things the lysosome can't eat up, and it will accumulate in lipofuscin. And you see that in old heart, for example, or old muscle. Now, a lysosome, if you have different steps, you have opportunity for disease. The eye cell disease or inclusion disease, there's no mannose-6-phosphate. In other words, there's no signal. There's no signal for it to go to the lysosome. So there's no enzymes. And what happens is the enzymes are secreted. They're produced, but then they are secreted constitutively. So they go out. You can find enzyme in an eye cell disease patient in the blood. So eye cell disease for inclusions. Remember, there's three structures. There's a membranous organelles, non-membranous organelles, inclusions. So inclusion is what they see because the autophagic and heterophagic components are not digested when there's no enzymes in the lysosome, and so they accumulate, inclusions accumulate in these cells. So in summary, lysosome has a host of enzymes, a single membrane, not a double, single, 50 enzymes or so inside there, safely contained in the membrane until they're not, like ischemia or something. Pathologic conditions can pop those. Primary lysosomes produced by Ruffyr and Golgi. Mannose-6-phosphate on the protein signals for protein sorting of the Golgi for the lysosome, and of course it is produced at a higher pH and then hydrogen ions pumped in for a low pH to occur. So cellular digestion, embryologic development, defense against microbial infection or functions, intracellular digestion of thing, autophagy. You got primary lysosome, secondary lysosome to occur, and maybe residual bodies or lipofuscin granules to remain. Smooth in the plasma reticulum, you see a network of vesicles in through there, and usually associated with lipid. Smooth ER. Smooth ER is produced by rough ER. So rough ER comes here pinching off as smooth in the plasma reticulum. Here you can see the smooth ER is pinched off of this. Here we can see here rough in the plasma reticulum here and here. And if you look at a higher mag, you can see that rough in the plasma reticulum are giving rise to smooth in the plasma reticulum that we see in these cells. Now, in terms of membranes and fluidity, you get proteins migrating around. They can flip, they can turn, but what they can't do is flip-flop. However, in the case of rough endoplasmic reticulum, the phospholipids are added to the outside, and then they're flipped over to the enzyme with a certain enzyme that allows that to occur, and that's how it's able to build a lipid bilayer, because they're added to the outside and then they're flipped over to the inside to make even lipid bilayers. So that's in the Golgi, rough endoplasmic Golgi and secretions. So here we can see these enzyme which allows it to flip it. Now the smooth endoplasmic reticulum also synthesizes and transports lipids in the intestine. And here we can see this intestinal absorptive cell. Here's the lumen where you got your food stuff and glycerol, fatty acid, and monoglycerides come through. And then they're resynthesized here as triglycerides. Then the triglycerides go through and interact with the rough endoplasmic reticulum that has pushed proteins to the Golgi. So from the Golgi, the resynthesized triglycerides pick up protein, and then they become chylomicrons, which are dumped out at the lateral borders of the cells, in between the different cells, and then they go into a lymphatic capillary or the central lacteal. So these are chylomicrons, as you can see, and you've got synthesis of a triglycerides in the smooth endoplasmic reticulum, and here's a central lacteal that we can say the intestinal villus, and these are central lacteals. So these would be central lacteals, and through there as well. If you look at individual cells, it's a simple columnar cells, as you see, and here you can just a hieromega one of these, and what we can see is you can see the lipid that's inside the smooth endoplasmic reticulum. So really, this is smooth endoplasmic reticulum that is resynthesized triglycerides that ultimately are going to discharge them as chylomicrons. So here we see electron microscopic view showing the lipids in the smooth endoplasmic reticulum in the apical portion of these cells up in this region. Now, also the smooth endoplasmic reticulum is involved in detoxification, like in the liver. And so of toxic things, toxin, what is a toxin? Toxicology is a study of poison. Poison substances are produced by plants, animals, and bacteria. Look at that poison ivy through there. This is anthrax, a bacterial toxin, and this is a little frog. Don't eat him, if you know what I mean. A toxicant is a specific poison chemical, xenobiotic, man-made substance, or produced but not necessarily found in the animal. The sting of a bee is a xenobiotic. The snake venom is a xenobiotic. It doesn't circulate in the animal, but it does produce in the animal. So we can see the sting of a bee, a xenobiotic, or associated with chemicals, sometimes synthesized by humans, but not in all cases. And so here you can see what's happening in the body. You get exposed to a xenobiotic, and then the body tries to store it, to to reduce the level, because it's a dose that hurts you. If it's too high a dose, it'll kill you. And then so it stores it in a bone, and that's where the problem was with the eagles with DDT, is that they would store the DDT, even though there's no more DDT around, it would still affect them because it was stored in their bodies. And so then you have action, you're trying to metabolize these products and try to excrete them. That's what the body's trying to do, pick up xenobiotics, work with them, try to excrete them. Basically, there are different phases. You have an oxidative phase, metabolites, and then you have a synthetic phase, a detoxification phase, where you try to reduce the size, to ionize it. You're trying to make it water soluble, excretable. When the doctor tells you to drink a lot of fluids, he or she is really telling you to excrete a lot of fluids in order to get rid of these xenobiotics. So also, smoothie are associated with steroid secretion, and here you can see where the smooth endoplasmic reticulum is associated with cholesterol production. Also, that goes in through, and then the final steps after the cholesterol, after cholesterol production is modified in the mitochondria, then it comes back through here again to produce the testosterone. Here we see a lytic cell of a horse with a bunch of smooth endoplasmic reticulum. There's a gap junction right there, but there's a host of smooth endoplasmic reticulum. There is a little bit of FER, but not much, and these are the mitochondria that we see. Also, the smooth endoplasmic reticulum is in muscle cells, and it has to do with regulation, calcium regulation of muscle contraction. And so here we see a transverse tubule, and this is a sarcoplasmic reticulum that's located. Sarcoplasmic reticulum is really the smooth endoplasmic reticulum. Here we can see it looks just like what we saw in the other cells, and it's going to hold the calcium. So smooth ER, structure and function summary, no ribosomes, a smooth surface, no ribophorins to bind it. It's synthesized triglycerides, cholesterol, and steroids is what it does. Has enzymes for conjugation, oxidation, methylation to metabolize detoxified drugs, and also sequester and release calcium in muscle contraction in the sarcoplasmic reticulum. That's what the SCR does. Now we move to mitochondria. Mitochondria, what they do is they convert sun energy stored in food. So the things we eat, plants, animals we eat, into a usable source, ATP, high energy bond, which is biosynthesis and movement. Here we see mitochondria over a period of time. They're not stagnant. They're always moving around. Inside the cell, we see some quite long in this case. Mitochondria can vary in length, but you see the dimensions there, mostly of them. Smooth contours is double membrane. Inner membrane, where you have Krebs cycle and different activities going on, and the outer membrane too. So there's two compartments. There's an intracrispy department inside here, and an intercrispy department. And the intra one is where you have the cytochrome oxidase, you have Krebs cycle, and ATP being produced in these. So a larger intercrystal space on the outside, and intracrystal space is on the inside where we have these. And here we see some mitochondria, several mitochondria located in that cell. Mitochondria move to where energy is needed. In this muscle cell, energy is needed, so it's right there around this flagellum. Again, energy is needed, and it moves to where energy is needed. Here again, we can see lots of mitochondria in through there, as they are associated with the intestinal absorptive cell for the ATP that's going to be needed for transport to occur. So this is the brush border, and here we can see these little globs in there are really mitochondria like we see there. Also, cardiac muscle cell, this is cardiac muscle cell, and you can see the mitochondria, the rods of mitochondria in there as well. Here again, they are right here. Many mitochondria needed. Now, in the Krebs cycle, we produce the ATP. So in between the outer and the inner membrane, you have a pH gradient. So that's where you have the hydrogen, the electron transport chain to have a higher pH, a lower pH on the outside, the inside, and that makes hydrogen ions want to come in. And when they come in, they change the ADP plus inorganic phosphate to ADP. So that's what they do. So by having this transport system, you're able to modify the pH and then modify the pH to help fuel these globular proteins on the inner membrane to produce ATP. Now, so cellular respiration, the process of organic compounds, glucose broken apart, releasing energy, ATP. And so here we can see it, glucose plus oxygen, enzyme, coenzymes yields CO2 and water, and release of energy and heat. And so that's what we do basically with respiration to occur. It's required oxygen to unlock the energy that's in food stuff, the energy from the sun that went to the plant that's being transferred to you through the food that we eat. You have to have both the food source and oxygen. It's why we are oxygen demanding animals. In contrast, a plant would take CO2 and use those to make the carbons in the plant itself. That uses the carbon source for plants are really from the air, from carbon dioxide. Very handy to have those that use carbon dioxide as well as those who use oxygen. But anyhow, that's a source of the heat too as well. So that's why we're warm-blooded. And here we can see a mitochondrium. Now, a lot of things can go in the outer membrane, but they can't go in the inner membrane. In order to get ATP out, you have an antiport. ATP goes out, ADP comes in. In order to bring in phosphate, you need that pH gradient. So the hydrogen ions come in with phosphate. Also, hydrogen ions come in with pyruvate. It goes through the outer membrane, but in the inner membrane, you have to have a symport, antiport, in order to get things to go inside. The inner membrane is less permeable than the outer membrane. And so, again, how about the other thing, oxygen, carbon dioxide, and water? Remember that those things, maybe there's a little channel full of water, but those things can just go through a membrane down its concentration gradient. If it's higher concentration in oxygen here, it's going to go out to the outside. So how about enzymes? Enzymes got to get in there too, and they're hard. And so remember, we have the signal of the protein that brings it to the rough endoplasmic reticulum. Also, that signal, that protein comes out from the cytosol, and it goes in through there, matches a receptor that's on this membrane, goes to the area where they're pinched, and now the protein is on the inside where it then is cleaved on the outside. So you have cytoplasmic proteins that are incorporated in there. What about proteins in between the two membranes? Well, here they have two signals, and one's cleaved on the inside, just like this, and that leaves a second signal, and then that one is incorporated into a channel here that ultimately that protein is cleaved off and released. So there's a way to get proteins in the inner membrane as well as the space between the membranes. So protein on the inner membranes are the transport proteins. They are ATPase synthetases. They are for the Krebs cycle too. So all those things are what has to go through into the inner membrane. Now, the function of mitochondria, one is for storage of calcium, and you can see calcium deposits in there oftentimes. Now, mitochondria have different shapes. Sometimes, like a hepatocyte, they have a lamellar shape. That's your typical mitochondria with the cristae or mellar shape. But in a steroid-secreting cell, there are a series of tubes like this. Astrocyte, they say they're like triangles. I've never seen that, but these are a series of tubes you can see. So this is the lamellar type that you have, like in the liver, and then these are the tube type, like you have in a steroid-secreting cell. Now, we mentioned that Smoothie R was involved in production of cholesterol, but when it's produced, the side chain is cleaved in the mitochondria, and then it comes back, and then smooth ER is involved again in actually final steps of producing testosterone. Now remember how the nucleus enfolded from a cell membrane to make a double membrane? Well, the proposed evolution of mitochondria is that a prokaryotic cell actually brought in the plasma membrane with it, and that this was the original plasma membrane of the cell, and that the membrane here is really the prokaryotic cell that became involved in synergistic. And here we see mitochondria. It doesn't come through the RER system. It basically it takes one to make one, and you get your mitochondria from your mother. And here you can see where a mitochondria is splitting into two. It takes one to make one, kind of like a cell. Mitochondria double membrane, and it has DNA. So you can see the DNA fragments, and the DNA that's in there is in circular form, like bacterial one. And here we can see it, the circular DNA right inside the mitochondria. So this is the mitochondria here, and you can see cytosolic proteins are incorporated. So they're incorporated with the transfer RNA. They're incorporated with the ribosomal RNA. They're incorporated with telling the cell to transcribe, as you can see, or even to replicate the DNA. So it's got circular DNA, and the cell regulates if you need more mitochondria to do that. If they do that, it get it bigger, and then they split. Is what this. And so these proteins here are all coming through that system. Ribosomes in the cytosol go into the mitochondria, and this is a scalar reaction that we talked about. The final component are inclusions. They're expendable inside the cell, need not be there for the cell to live, very important for the cell function. And one of those, we see glycogen in this platelet, or you see lipid droplets here. Those are examples of nutrients, which are one of the three types of inclusions. Another type is secretory granules. And here we can see, this is the patent cells, secretory granules, and you can see the secretions here of the goblet cells, and this is like a pancreatic astrocyte secretions. So these secretory granules are inclusions. Need not be there for the cell to live, but it is important for the cell function. Another group of inclusions are the pigmentation. So like melanin granules, they're inclusions too. Here we see lipofuscin in old muscle. A lipofuscin is lysosomal degradation as far as it goes, and then it can't get it all destroyed, and so you have some remaining as lipofuscin. So inclusions, expendable. Here we see lipofuscin in through here. The eye cell disease is for inclusions. Remember, the enzymes are released constitutively and don't end up, because there's no mannose-6-phosphate, they don't end up in the lysosome as they should. No mannose-6-phosphate, no signal for enzymes to go into there. Next time, we're going to talk about transport endocytosis or phagocytosis, or go to Colorado. Boy, it was a cold day then. I had snow chains on my Triumph. It was something. And here we are in Yellowstone National Park on a very snow day. There was about 12 feet of snow. That time I was a real cold rascal at that day. And here we enjoyed the hot spring. This is not me. This is my friend. I took this picture. But we enjoyed the hot springs in those days.
Thank you.